Hi, we're ProteinVision. In this video we will show how protein language models can be used to predict protein mutation or variant effects. We will cover two settings. First, how to use a pre-trained protein language model as a zero-shot predictor for mutation effects. This means we use the model directly without retraining it for a specific protein. Second, how to fine-tune a protein language model when experimental data is available. If you are new to protein language models, I recommend first watching our introductory videos. Here we will start with a short recap of the main idea. A protein language model takes an amino acid sequence as input. First, it converts each amino acid into a numerical embedding. These embeddings encode both the amino acid identity and its position within the sequence. Then the model repeatedly updates these embeddings using the attention mechanism. This allows each amino acid embedding to incorporate information from the other positions in the sequence, in particular from amino acids that are close in the folded 3D structure. After several layers, we obtain a context-aware embedding for each position in the protein. These embeddings contain information not only about which amino acid is present, but also about the structural and functional context of that position. Most protein language models are trained with mask language modeling. During training, we randomly mask around 10 to 15% of the amino acids in a sequence. The model then has to predict which amino acid belongs to each masked position. The model outputs for each masked position a probability score for all 20 possible amino acids. So the model learns a very useful question. Given the rest of the sequence, which amino acids are plausible at that position? This is already very close to mutation effect prediction. If we want to know whether a substitution at one position is likely to be favorable or unfavorable, we can ask the model, how likely is the wild-type amino acid at that position, and how likely is the mutant amino acid? Let us assume we have a wild-type protein sequence and we want to evaluate a mutation, for example, at position 4, a mutation from the amino acid methionine to the amino acid alanine. To do this with a pre-trained protein language model, we mask that position in the sequence. We then pass the masked sequence through the model and obtain predicted probabilities for all 20 amino acids at that position. So the model gives us a probability for the wild-type amino acid, a probability for the mutant amino acid, and probabilities for all other amino acids as well. These probability scores for all 20 amino acids sum up to 1. A simple and commonly used score is the log probability ratio. We divide the predicted probability for the mutant by the probability of the wild type, and we then compute the logarithm of the resulting value. This compares how strongly the model prefers the mutant amino acid over the original one. We can interpret the resulting score as follows. If the score is greater than 0, the model assigns a higher probability to the mutant than to the wild type. If the score is smaller than 0, the model prefers the wild type. And the score close to 0 means the model sees both amino acids as similarly plausible. So the sign of the score tells us whether the mutation looks favorable or unfavorable according to the model. Why does this make sense? During training, the model has seen millions of protein sequences and learned which amino acids are usually found in different sequence contexts. If a position is strongly constrained, the model has mostly seen the same amino acid or only a few similar ones at that position in related proteins. So it will assign high probability only to a small set of amino acids, while many unlikely substitutions get a lower score. If a position is more flexible, several amino acids may be plausible there, and the model assigns relatively high probability to several different amino acids. So this score can be understood as a measure of how well a mutation fits the learned grammar of proteins. As we will see in the next video, these scores often correlate well with experimental measurements such as activity, stability, or function. At the same time, the score is not a direct measurement of one specific property. It is better viewed as a general plausibility or fitness proxy learned from natural protein sequences. We can apply the same idea to variants with multiple mutations. For several mutated positions, we compute a score contribution for each mutation and then we add them. So why do we add these values? Because logarithms turn products into sums. So adding the individual log ratios is equivalent to multiplying the probability ratios across positions. This gives us a simple combined score for the full variant. In this example, we see that we compute the log probability scores for both masked positions, and then we simply add the two scores. Intuitively, each mutation contributes evidence for whether the variant is more or less plausible than the wild-type sequence, and the total score combines these contributions. Again, the interpretation is greater than 0, the model overall prefers the mutant combination, and smaller than 0, the model overall prefers the wild type. One important note is that this is an approximation. Interactions between mutations can be complex, and multi-mutation effects are not always purely additive. Still, this additive score is widely used in practice and often works surprisingly well as a first estimate. So far, we used the protein language model in a zero-shot way. This means we use the pre-trained model directly to estimate general mutation fitness, without training it on a specific experimental dataset. But if we have experimental data for one protein or for a protein family, we can do more. If we have a dataset with measured properties for many mutants, we can fine-tune the protein language model to predict a specific property of interest. For example, this property can be enzyme activity, thermostability, binding affinity, or another experimentally measured function. This is an important difference from zero-shot prediction. In the zero-shot setting, the model gives a general score related to how plausible or fit a mutation looks based on patterns learned from natural protein sequences. With fine-tuning, we adapt the model to predict one specific experimental property. The setup is similar to standard fine-tuning for protein language models. We take the amino acid sequence of each mutant and use it as input to the model. Importantly, in this case, we do not use masking during training or prediction. We simply provide the full mutated sequence. We then use a classification token, often called a CLS token. This token is updated together with all the amino acid embeddings as the sequence passes through the model. After the final layer, we extract the CLS embedding and use it as a compact representation of the full mutant sequence. This representation is then passed to a small neural network, for example a multi-layer perceptron, which predicts the experimental value for that mutant. We train the full model end-to-end using the experimental measurements in the dataset. So during training, the model learns to connect sequence changes to the property we care about. After fine-tuning, we can use the model to predict the same property for new mutants that were not part of the training data. This allows us to move from a general fitness estimate to a property-specific predictor tailored to the protein and dataset of interest. In the next video, we will implement zero-shot prediction and fine-tuning for mutation or variant effects in Python, and we will apply it to a dataset containing many experimentally measured mutants.